Hi everyone. Now we are going to discuss a topic called microorganisms as food. Yes, microorganisms as food. We are directly consuming some microorganisms as food. Examples are single cell protein, single cell protein and mushroom. So mushrooms, we already discussed this in another video I uploaded, so go through it once. Okay, in this part we will discuss about the single cell protein. So let's go into the topic. So the conventional sources of protein. Quantitatively, proteins occupy the second position in our diet after carbohydrates, that we know. Proteins play an important role in many life processes. So conventional sources of protein like some plant proteins and some animal proteins like pulses, groundnut, beans, cereals, plants, meat, fish, eggs, milk and cheese or animal proteins are produced in on very large scale, but still it's unable to meet the requirements of the increasing population. In view of this, search for non-conventional source of protein had begun long back, but a promising and potential source of protein called single-cell protein has been found to supplement human food and animal feed from the last few decades, that is approximately of 1960s. Okay, so let's see what is a single cell protein, how it is going to be produced, and what are the advantages of the single cell production, and then what is the applications of this single cell protein. So single cell protein, the term single cell protein refers to the total protein extracted from the pure cultures of microorganisms. So what are microorganisms? It may be an bacteria, fungi, that is mainly filamentous fungi, then yeast, algae, actinomycetes. So these are all going to be considered as microorganisms which are widely used in the production of single cell protein and can be used as a protein rich food supplements by humans and animals. So both humans as food and animals as a feeding. So that kind of cells or cell biomass is being termed as single cell protein. So this single cell protein is also being called as microbial protein or novel food or mini food. So actually this single cell protein term was coined by Mr. C.L. Wilson in 1906. According to him, the single cell protein are the cells of algae, bacteria, yeast and fungi grown for their protein contents. That means he defined that single cell proteins as the cells of algae, bacteria, yeast and fungi are grown for their protein contents. He termed like this as a single cell protein definition. Then coming to what are the advantages of this single cell protein? So we have so many advantages. So let's see. In addition to protein, single cell protein also contains carbohydrates, minerals, lipids and vitamins. And according to the IU-PAC, so what do you mean by IU-PAC? International Union of Pure and Applied Chemists recommended, instead of calling it as single cell protein, better to call it as single cell biomass, that is proteinaceous single cell biomass. So as I told you, this nutritional values are good for protein. Along with that, it is also going to have the carbohydrates, lipids, protein, minerals and vitamins. So their nutritional values are as good as the conventional foods. That means we are having some conventional foods like pulses, grains, cereals, and coming to animal proteins like fish, milk, cheese, all these things. So it is equally having the same nutritional value. That's the one of the advantage of SCP. Then coming to microorganisms has have very fast growth rate when compared to our normal plants and the animals. So obviously we'll get the more food in the less time. Then we are going to have the example you can take algae will grow within 2 to 6 hours, and coming to yeast it will grow to 1 to 3 hours. Coming to our bacteria, it is of half an hour to 2 hours. Okay, so this is how, as they are having a very rapid succession of generations, so we can cultivate a large quantity of the protein in a less time. Then coming to the third point, protein content is quite high. So as I told you, it's having the nutritional values similar to the conventional foods. Still, it is going to have a high protein content when compared to a conventional. It may be varying of about 43 to 85%. They can easily grown on varieties of raw substances, including waste of agriculture, urban and industrial, and thus it is going to have the another advantage of removal of pollutants from our environment. So these are the few advantages of the single cell protein that made us to have the attention on the production process. So let's see what are the organisms that are involved in producing the SCP. So here are the list of microorganisms used for production process, as we already learned about the this microorganisms in the definition. So we are having the variety of microorganisms represented in here. We'll go with the bacteria first. Okay, so bacteria, as I told you, Pseudomonas, Methanomonas, and here Lactobacillus, Bacillus megacheria. So these are all involved in the production of single cell protein. Along with that, I already told you one more is actinomycetes, where we are having a nocardia, streptomyces, and so on. You can go on with the examples. Then coming to yeast, so here is your yeast, where Saccharomyces cerevisiae, Candida torticollis, and Candida utilis, Torulopsis. Okay, so these are all the few examples that we can write. Write under the single cell producing organisms. Then coming to the fungi or filamentous fungi, so you're having the Mucar, Rhizopus, Aspergillus species, that is Fumigatus, and Nizer, etc. So uh, algae is going to be the next important one, where you're having the chlorella, spirulina. Chlorella example is Senegasmus, and spirulina is going to be of a best example of blue-green algae, whereas chlorella is a green algae. Okay, and Senegasmus is also a green algae. So that's how we are going to have these list of single cell producing microorganisms. Sorry, single cell protein producing microorganisms. So let's have a quick review. So we are having the bacteria, then you are going to have the actinomycetes, yeast, filamentous fungi, and algae. Here I didn't mention the actinomycetes, so you can write it actinomycetes also. Okay, so you have to remember, and these are very important to write in the answer. Then coming to the comparison of the single cell proteins in different organisms. So which one is the best if you want to opt? So here are the list of the protein percentage that is being produced by these organisms. So yeast will be of nearly 45 to 55%, and then bacteria is about 50 to 80%. And we know that the protein content is quite high, and it is going to have a very fast growth rate, so 50 to 85%. Then filamentous fungi is only 30 to 35%, and algae is 45 to 65. So out of all these things, majority of the single cell protein production is going to be occur mainly on bacteria, yeast, and then algae, and the last place goes to the filamentous fungi. So this is all about a overview of the protein percentage of these organisms. Okay, then here are the raw materials. The next concept that we are going to discuss is the raw materials. A variety of raw materials are being utilized for the production of single cell protein. So these includes the waste formed in our urban areas, agriculture, and industrial. So here are few raw materials and their uh, microorganisms on which they are being. For example, when you take the bacteria, so here are a list of bacteria. Say take Pseudomonas, I told you. So here this Pseudomonas is going to be grown on a substrate called as glucose, uric acid, and other non-protein part, then nitrogenous compounds. Okay, so these all can be used for the growth of the Pseudomonas fluorescens. Then coming to your methylomonas or methanol. So methanol is the main component or substance on which they are being cultivated for this production. And then coming to the fungi, as we know the examples rhizopus, mucor, aspergillus, all these things. So aspergillus fumigant, and they are being cultivated on maltose and glucose, whereas aspergillus niger and oryza is being cultivated on cellulose, hemicellulose, all these things. Okay, and then coming to yeast, that is candida, torulopsis, saccharomyces. Obviously candida is going to be of glucose compounds, and then candida novels is going to be grown on N-alkanes, and Saccharomyces cerevisiae on lactose, pentose, and maltose. Then coming to the algae, so we are having the chlorella spirulina. Okay, so the spirulina is mainly going on the photosynthesis, carbon dioxide. So algae is majorly dependent on the sewage water where you can cultivate, that I'll show you. So here the for the algae the substrate required is the water. So the other thing, carbon dioxide is going to be available from the atmosphere, so it can utilize and it can be grown. So these are the few raw materials that are necessary for the single cell production. Then this is what I told you, that this is all the algal biomass where it is cultivated in sewage oxidation ponds. In the sewage plant, we are having the oxidation ponds where they are growing this one, and they will collect all this algae and they will process it, and that is going to be used as a single cell protein. And this is what the bacterial and fungal biomass is being produced from the sewage plant or wastewater. Sorry, wastewater. And then this is the yeast biomass where in the fermenter tanks these are being produced in a large quantity. Okay, then coming to the production step. Okay, this production mainly in India is being taken up by two institutes, big institutes. One is the NBRI and another one is the FTRI. NBRI means National Botanical Research Institute, and another one is the CFTRI is Central Food Technological Institute. In these institutes, algae cultured on the sewage, okay, wastewater. Now coming to the production steps of single cell protein, it involves the following steps. The first one is the selection of strain, that is microbes, and then selection of the substrates, then fermentation process, then harvesting obviously, then post-harvesting treatment and processing of single cell protein. So these are the steps that we are going to discuss in detail one by one. So now coming to the selection of microorganisms or microbes and substrate. So this step is very crucial step, and find the selection of microorganisms is mainly depend upon the available raw materials. And remember that the quality of the protein, that is single cell protein, also depends upon the selected strain. The strain is nothing but the microorganism that you are taking. Okay, for instance, hydrocarbons are being used as a raw material for the cultivation of bacteria, yeast, and the fungi. Then agricultural waste is going to be having as a substrate for mainly for the fungi, as it contains the more cellulose and hemicellulose components. Okay, and care should be taken that the selected strain should not produce, so it should not produce any toxic or undesirable effects in the consumer. This is very foremost thing that we have to keep in mind, that it should not cause any toxic or undesirable effects in the consumers. Okay, so that's how you are supposed to select the organism as well as the substrate. Then coming to the fermentation process, that how you are going to have the, so there are majorly two types and sometimes three are going to be used. So two types of processes which are used for the production of SCP, namely submerged fermentation and semi-solid fermentation. These are the two types of the fermentation mainly used, and this fed batch is mainly used for the culture of what we call microorganisms are cultured in this fed batch culture system. But our single cell production will occur in either the submerged or the semi-solid fermentation. What is this submerged? Submerged type of fermentation process is always a liquid. That means it occurs in a liquid condition. Semi-solid, obviously the name indicating it's of a semi-solid. So then coming to the harvesting, So once the fermentation completes, how it is going to have the harvesting. When the micro colonies or when the colonies of microbial cells are fully developed, they are then harvested. The bulk of cells are removed from the fermenter by a process called as decantation. By a process called as decantation. That means you are just removing by the decantation process. Now, after decantation, what we should? So now it was harvested, but we can't use it. So we have to have some sort of a post-harvesting processes. So what are those things? We'll discuss. See here a rough diagram. A substrate and the nutrients and the microorganisms are added in the fermenter, and the fermenter is a vessel where you're supposed to have your incubation and the growth of the organisms. Okay, now once the organisms are fully grown, it will be filtered and it is going to dry, and that's how you are getting the single cell protein. Sometimes some cells may directly get died without the filtration. So we may have direct drying or through the filtration and this process. So this is all the harvesting process. Filtration, drying, everything. Okay. So as I told you, we are having the two types of fermentation process. One is a submerged, always in a liquid condition, and the semi-solid fermentation. Now let's go into the post-harvesting process, how it is going to occur after the decantation. Separation by centrifugation, heating and drying, etc. So you are going to have the post-harvesting treatment. After harvesting, the cells are subjected to a variety of processes such as heating, drying, and centrifugation. Okay. Now the processing of single cell protein. So we had seen that you separated and you are ready with the harvesting. Now it should be of some processing to remove its smell or unwanted material, anything. It should be processed. So how it is going to be processed? So let's see. So processing of single cell protein is done by mainly two methods. One is a liberation method and another one is a reduction method. So what do you mean by liberation method? Here in the liberation, the cells are destructed uh, by the some mechanical methods or by chemical methods or by some physical methods for the indigestible cell wall. So here liberation of cells by destruction of indigestible cell wall by all these methods. So what are all these methods? Mechanical methods. The mechanical methods includes crushing, crumbling or grinding. So by all these things we can break the cell wall of those things and we can get it, and even with pressurized homogenization also. So these all comes under the mechanical methods. Then coming to the chemical methods, where you are going to use the enzymes or salts to digest or disrupt the cell walls. So for example, salts like NaCl, sodium diadecyl sulfate, or enzymes like nucleases are also being used in the chemical methods to digest the cell wall. Then coming to the physical methods, the best is physical methods are freeze thawing, then osmotic shock, heating and drying comes under the physical methods. So by these methods, we are going to disrupt the cell wall and liberate the protein from the cells. Then coming to the reduction, here nucleic acids can be removed by a process called as alkaline hydrolysis, or by acid precipitation, or by exogenous and endogenous nucleases. It is also necessary that we should remove the other contents which are going to have some sort of a negative effect. So mainly of nucleic acids. So along with the protein, nucleic acids are also present in the single cell producing organism. So we are going to lyse that nucleic acid by using an enzyme called as nucleases. Okay, so here if you go through just the overall production, so here is the substrate and then they are being kept in the fermenter and you are using the nutrients and the microorganisms in the fermenter, and then that is a culture is being added, and incubation is going to, and the fermentation process completes, you are going to filter it and then you are going to dry, and then you are going to have the single cell protein. So this comes under the processing, and this is going to be the harvesting. Okay, so these are the about your production of processing of single cell protein. So we finished off all the steps of production steps. One is the selection of strain of microorganisms and substrates, then fermentation process, two types, and then harvesting, So post harvesting things also we had seen, and the processing of single cell protein by decantation process and by liberation, harvesting by the decantation process and processing by the two methods. One is the liberation and second one is the reduction process. So let's see about the applications. This is the overall process of our uh, production process of single cell protein. This is an example of spirulina production. Okay, so these all steps indicates the cultivation, that means your first selection of strain, everything, and then the harvesting step, then how you are extracting and what are the end products of the concerned thing. So here I showed you the spirulina as an example. Okay, so what are the substrate that we require is water, algae, carbon dioxide and nutrients, obviously. So they can be cultivated in open ponds that I showed you, greenhouse, closed ponds, photobioreactor, and other closed systems can be used for the cultivation of this spirulina. Then after that you are going to have the harvesting process, either by drying, centrifugation, flocculation, filtering, all the things are being used in the harvesting process. Then you have to dry it. After drying, it have to undergo further processing. That processing is nothing but extraction, either by the chemical methods or by the mechanical methods. Okay, so from that again it is being refined, and that refining may be of having so many end products which are going to be of listed here. It can be of biodiesel or Z fuel, ethanol, hydrogen, methane, like this we are going to have so many end products from this one. So now we will discuss about the applications of uh, this single cell protein. So here are the products, the marketed products of single cell protein, how they are going to look like. This is just an example how they will be available in the market. Okay, so as a source of protein, we know the a single cell protein is a good source of protein, and it also possesses various vitamins, amino acids and minerals, and it is going to be a good food supplement for undernourished children, and it has a cholesterol lowering effect, that means it can control our obesity. So as it is having the lowering of cholesterol effect, so it reduces the body weight, isn't it? And not only that, it also provides instant energy. Okay, and another important thing, it reduces the premenstrual stress, and it lowers the blood sugar in diabetic patients, and that lowering is due to the presence of beta linoleic acid that is present in the single cell protein. Then it contains beta carotene, which is very good for skin, eyes, and not only that, also reduces the cancer risks. Okay, then phycocyanin is another component of the single cell protein, which stimulates the immune system, providing protection from several diseases. Then it is being used in several cosmetics also. Then single cell protein used to feed cattle, fishes, etc., and these are also being used in the poultry and animal feeding industry. And along with all this single cell protein are also having some medicinal uses. So these are all the applications of some of the applications of single cell protein. Still we are having a more advantages, and that's why it is being having a very importance in the research field of producing this one to uh, what we tell is to maintain the or to cater the increasing population, to cater the protein content to all the increasing population. Okay, and then there are certain some disadvantages also, some may be allergic to this single cell protein, and some may have the indigestion problem, and some may get the kidney stones, and etc. are the some of the side effects, disadvantages of this single cell protein. But still we can neglect when we compare to the more advantages of this single cell protein. Okay, so by now you may come to know about the single cell producing organisms and production process, what are the advantages, applications, all the things. Okay, so this we finishes the microorganisms as a food. One of the part is single cell protein, other part is mushroom, where you can go through in another video that we already had it. Okay, thank you.